Okay, so one of the objectives says to be able to identify the type of hypersensitivity for each of the following. So these are examples, right, of particular hypersensitivities. And as I said on the test, they'll be written out just like this, right? Type 1, immediate IgE mediated. Type 2, cytotoxic. Type 3 is immune complex mediated. Type 4 is delayed cell mediated. So serum sickness we talked about last time, and I was a little bit wrong in some of my information. Shame on me. So I'll correct that today. Anybody remember which one of these hypersensitivities it was? It's three. So immune complex mediated. And this is when you get injected with antibodies. Sometimes your body will make antibodies against those foreign antibodies. The example I gave was they used to commonly give horse antibodies for snake bites. And this is true. And what is that reaction happens about 7 to 14 days later when the person makes antibodies against it, and that antibody is still maybe in their system. So it can happen upon first exposure, but again, it's going to be that delayed time frame, the time it takes for your body to make an antibody against it. So I did some research because in the past, as I said, there was, you ran into difficulties if you gave them that same serum again, if they had gotten bit by a snake again. So I did a little bit of research, not a ton, but they have done some things to the antivenomins now, even though they make them in other animals and still. They're less likely to cause this, what we refer to as a serum sickness, okay? Serum sickness would sometimes happen too when individuals have tetanus or diphtheria. One of the treatments we could give is give them antibodies from a horse. Nowadays, what they do is they actually take from your blood, when you donate blood to the blood center, they can take lots of different things from that. And one of the things they'll tend to do is they're going to take antibodies out of your blood and purify them to be able to give them as treatments for people with these types of diseases, right, that might have diphtheria or tetanus, where they could give them the antibodies before their body could make it to help them, because the toxins for diphtheria and tetanus, as we're going to talk about later, are very fast acting and they're very detrimental to the body. So the TB skin test we haven't talked about yet. Does anybody know which type of hypersensitivity this one is? So we haven't talked about it yet. So it's the number 4, right? Delayed cell mediated. So we're going to go over that one in a moment. Hives, do you guys remember which reaction this would be?
Would it be the IgE?
Yeah, it's IgE. It's number 1, IgE mediated. Asthma, type 1 as well. Transfusion reactions, 2, right? Cytotoxic, we're dealing with cells. Hemolytic disease of the newborn? Nope, also 2. Also 2. All the blood-related ones are cytotoxic, right? Think of the blood cells, right, being destroyed. It's type 2. So let's go back up to type 4. So for type 4, this is the only one we're not going to talk about antibodies. Right? All the others we talked about antibodies, didn't we? This one is, as it says, cell-mediated, and it's delayed because of that. And the tuberculin test, right, is an example of that type of reaction. Takes advantage of this. So I am actually going to play this one. A good thing I already turned down the volume earlier. So I'm going to go ahead and play this one.
Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TB cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen the second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
So just like all the other reactions we've been talking about, this is a normal immunological reaction. The problem is, right, it's not necessarily something that would necessarily harm us, okay? This particular example of poison oak, okay? It's just how our body is reacting to it, okay? So the normal steps, right? Antigen presentation, cells, T cells being activated, right? Second exposure, T cells are going to communicate that this bad guy, according to them, right, has gotten back in. Infiltration is the most common, inflammation is the most common immunological response, right? And so in this case, this is all cell-mediated, right? We didn't talk anything about antibodies. Where in all the other ones, right, it was antibodies that were setting off this reaction of inflammation. So as we saw, this develops slowly because you need the cells to infiltrate into the area. And this can happen just about anywhere. So contact dermatitis, tissue damage, sometimes this will happen. And this will sometimes be the reason why our tissue grafts or organ transplants will fail, is because the helper T cells are recognizing the foreign tissue as being foreign, right? And sending macrophages in to attack. Sometimes autoimmune disease, again, where our T cells are recognizing ourselves as self and destroying them. But the one we tend to focus the most on, of course, is the TB. But to give you an example of some other types of, other than poison oak, also of course poison ivy, some people are allergic to nickel. Nickel is very common metal used in what's referred to as costume jewelry, right? So the stuff you buy at Claire's and stuff like that tends to be nickel and plated in gold or silver. So when that plating wears off, then people's skin comes in contact with it and they can have a serious reaction. So I have an aunt that's allergic to nickel, and you know, she's like, well, that just means he has to buy me the good stuff. Right? But you know, if you're having a reaction, it's probably that's the case. Chromium salts that are used in curing leather can also be allergic type reaction for some people. Latex products can have this type of reaction for some people, where it's cell-mediated, but some people are also type 1 IgE mediated, and again, usually that reaction is going to happen immediately, where with the delayed, of course, you know, it'll be a little bit later, and you might question, well, why am I having this reaction? What did I come in contact with? Right. And then for some people, as far as latex is concerned, they actually can have both of these types of reactions, and then it's really severe. But thankfully, right, lots of places have I've gone away from using latex gloves. So the tuberculin reaction is where they purposely put proteins associated with tuberculosis in your skin. And this is testing to see if you've been sensitized, right? Does your immune system recognize this? Has it seen it before? Okay? So if you have the disease, and this disease is very slow progressing, so this is one of the early tests that we could do to indicate if you have this infection, right? But the problem with this is that if you've been vaccinated, is your body going to respond? If you've been vaccinated. If you've been vaccinated, you've been given, right, usually a weakened form, or in this case, a host relative to Mycobacteria tuberculosis. It's Mycobacteria bovis, I think they use. So it's not a very good vaccine. It's one of the main reasons why we don't use it very often in the United States. But it is used in other countries, and it is used in the United States under certain circumstances. Military, sometimes when they have to go overseas, will be vaccinated. Unfortunately, it's a really bad problem in the prison system, so a lot of the prison guards and workers have been vaccinated against this. Those of you guys going into the health field, you may also experience getting the TB test a lot, so that many years down the line, you might end up with a positive TB test, because basically them giving you this protein so many times, eventually your body may mount an immunological response that it remembers it. So it's almost like you've been vaccinated, okay? So this sometimes happens. Once you're positive, you'll always be positive, right? Once these cells recognize it, they're always gonna come whenever you get this shot, whenever they do the TB test on you, right? And it tends to be a hard induration, right? Because it's cells infiltrating into the area and macrophages. So don't freak out, right, if later on you end up with a positive test potentially, right? It doesn't necessarily mean you have the disease. It just means your immune system is recognizing it, okay? So, you know, like I said, there are people that come from other countries, right, and have been vaccinated, and maybe they don't even realize it, and they end up with a positive TB test, and, you know, healthcare professionals freak out because they haven't been properly informed, right? That this does not mean you have the disease, right? It means that your immune system is recognizing it. Could mean you have the disease, right? But not always. Once positive, always positive, right? Once your immune system is recognizing it, will continue to recognize it. Does that make sense to everybody? So that's an important point, especially for those of you guys going into the health field. So the other thing is properly administering the shot. My husband, when he worked for the prison and when he was sent overseas, he had been vaccinated twice for tuberculosis. When he went to go to LSU for graduate school, he gave the girl, I kid you not, like 5 pages of vaccinations. She's like, you have to have the TB test done. He's like, it's going to be positive. She freaks out. He said, I just handed you 5 sheets of vaccinations. It's on there twice. I've been vaccinated twice. Pretty good chance it's going to come out positive. She's like, well, you still have to have it done. He's like, okay, fine. So they inject it. A few days later, he's like, I don't understand. It's not positive. I'm like, I don't understand either. You've been vaccinated twice. What are the chances of that it didn't work both times, right? And I was like, well, how did she administer the test? I'm like, did she have to drop a syringe from a vial? And he's like, yeah. I'm like, did you see her shake the vial? He's like, no, I don't think so. I'm like, it's a protein in a suspension of saline usually. It'll settle out a solution. If you don't shake the vial, chances are you're not going to pick up any of the proteins. So she probably just gave him a saline shot, which he was happy because he should have been darn positive, which means the next step is what? To confirm you don't have tuberculosis. What are they going to do? They're going to do a chest x-ray. They're going to do a chest x-ray, right? So those of you that end up with positive TBs on a regular basis every year or so, right, they'll be having to send you for chest x-rays, right, to make sure you don't have tuberculosis. Okay, I told him he had to go report her for not properly following protocol. Whether he did or not, I don't remember. It was quite some time ago. So we already went over this one. This was that slide that I augmented just so that we could go over which ones I want you to be able to match up to the different hypersensitivities, the different examples we've talked about. So the next is, right, this is some information that we've kind of already gone over, but other is probably relatively new. So as we compare and contrast each of these hypersensitivities, we're going to look at similarities and differences as it relates to the cell type that's responsible for the hypersensitivity, the type of antigen and antibody. So there's a table from your book, and it's a pretty good table, but it's got some extra information, and in some cases a little more detail than I want you guys to know. So I made my own table that I'll post for you guys today. So very similar to that, and I kind of changed the order a little bit because of the way that I wanted to discuss it. So the type of antigen we didn't really talk about very specifically. They refer to them as being soluble or cell-bound. Okay, which one of these did we talk about that it actually would be a cell that we're attacking and killing? The clue is in the name of the hypersensitivity. I even left off the 1, 2, 3, and 4, but they're in order. Which one deals with cells? Delayed cell mediated, yep. It has cell in its name. There's one more. Cytotoxic, right? So for IgE, they're what we refer to as soluble antigens that are going to cause this type of reaction, meaning that, you know, they could be dissolved in solution. They're not very large. For cytotoxic, because we're killing cells, obviously it is a cell that the antigen is bound to, right? So this is our red blood cells, right, with their antigens on their surface. For immune complex mediated, these are again soluble antigens. For delay cell mediated, as the name implies, right, cells are involved in the reaction, but they could also attack cells, right? Because remember we talked about, and I mentioned, tissue and organ transplants, right, recognizing cells as being foreign. But in the case of things like poison oak and nickel allergies, these are all soluble. So it can be either or for this one. Now, the answer to this one is in the name of the hypersensitivity, so you better not get this one wrong. What type of antibody class is involved in immediate IgE-mediated hypersensitivity? What does the title say, y'all? IgE. IgE. The answer is in the title, right? IgE. But not for cytotoxic, but it is antibodies, right? What are our two main player of antibodies inside our bodies? Two that we talk about the most. IgM and IgG. So it can be either one or both, right, that are causing this, right? So I made that distinction here. It could be IgG and IgM, or it could be just IgG or just IgM. Immune complex mediated, right? So immune complexes, remember, are antigen and antibody. So we already know it's a soluble antigen. What type of antibody is it probably? The most common one we talk about. IgG. Delayed cell mediated. Do we talk anything about antibodies? No. No antibodies. This is cell mediated. Cell type responsible. So in other words, where did the IgE come from? Who makes IgE? What white blood cells specifically make antibodies? More specific. B cells, right? And if you want to be very specific, it would actually be the plasma cells, right? Right, but we're going to stick to B cells. So what's the next answer? What cells make IgG and IgM? B cells. One more time, what cells make IgG? B cells. Okay, this one's different though, right? Delayed cell mediated, who did we just say that was responsible? What's the cell that's responsible for recognizing that these substances or cells are foreign? T cells. Okay. Other cells involved. So, IgE, produced by the B cell, but then what is it attached to? What cells? Its Fc portion of specifically IgE attaches to a specific white blood cell found in our tissues and in our blood at a very low level. Basophils and mast cells. The mast cells are the ones in your tissues. Now, for cytotoxic, right, we've got IgM and IgG binding to these foreign red blood cells, right? Who's going to come along? Well, if it's IgG, it could be a natural killer cell, right? Because remember, they can recognize IgG attached to cells and it'll kill that cell. That process, abbreviated here ADCC, but what does it stand for? A is for, how does that natural killer cell know to kill that cell? What's on that cell? What's IgG? It's an antibody, right? Antibody-dependent cellular cytotoxicity. Right? It's a very special, unique outcome that can happen when IgG binds and the natural killer cell recognizes that Fc portion and kills that cell. With an immune complex mediated, we're going to keep it to just white blood cells, right? Again, neutrophils, macrophages, so it's a large group, right? But in general, our white blood cells are responding to antigen antibody complexes, what we call an immune complex. For delayed cell mediated, again, we'll just say white blood cells. As they said, macrophages, T cells are going to come into the area. Mediators, so these are chemicals that are released. There's one specific mediator that we talked about that I want you guys to remember for mast cells and basophils. It's a major contributor to allergy. Histamine. Histamine is the one. All right, it's going to cause that inflammation in that area. Now, IgG and IgM, when they bind to these red blood cells, IgG we know can cause the death by natural killer cells, but when IgM binds, how is it that these cells end up getting killed? No, because an antibody just binds to the surface, right, and alerts our immune system, opsonizes it for destruction. But what other molecular things, what other proteins are present in our body that can interact with IgM and IgG and cause the death of those cells by cell lysis? By the MAC complex, actually, the membrane attack complex. What system has the membrane attack complex? Starts with a C. Complement. Complement. Right? Is one of the major mediators, right, of this type of reaction. Immune complex. Can complement get involved with that too? We're talking about IgG, right? Absolutely. Complement will be one of the main reasons for the inflammation in those areas where these small complexes of antigen and antibody collect. How do white blood cells talk to each other? What's the general name we give to those chemical signals that they share with one another? Cytokines, right? So cytokines are the mediators here when you have delayed cell mediated. How much time does it take for immediate IgE mediated hypersensitivity to happen? It's immediate. What's immediate mean to you guys?
Right now.
Right now, seconds.
Yeah.
Seconds. It can happen in seconds and take as long as 30 minutes for the reaction to happen. But we're talking about immediate, fast. Right? So it could be as fast as seconds, right? but it could take as long as about 30 minutes, but that's still pretty fast. Cytotoxics, on the other hand, from hours to days. Same thing with immune complex mediated, right? And then what did we say was the key time frame for that peak for delayed hypersensitivity?
48 hours.
48 to 72 hours, right? And that's why the tuberculin test is usually read 2 to 3 days later, right? Because that's how long it's typically going to take for that reaction. Can you have a skin reaction with IgE mediated, immediate IgA mediated? Can you have a skin reaction? Yes. What's the common name we usually give to this? Hives. Hives, right? Or a wheal and flare reaction. Of course, this doesn't always happen, right? Asthma is an example of IgE mediated, right? You're not having a skin reaction there, you're having a reaction in your lungs, right? With certain injected, right, or ingested things, you won't necessarily have a skin reaction. You might have a system-wide reaction where you go into shock, right, anaphylactic shock. So this isn't an always thing, but it can happen. Can you have a skin reaction when you have a cytotoxic? And what do we think, what is the example that we think of when we think of cytotoxic? What's happening? Blood transfusion reactions, right? Or hemolytic disease of the newborn. Do you have a skin reaction with that? No. That's internal, right? Immune complex mediated, right? So if, say, this reaction is happening in your skin, are you going to have a skin reaction? Yeah, right? We refer to this particular type as arthros, right? Where they've injected it in. And remember I told you guys about how sometimes you'll have that reaction if they give you the tetanus vaccine too soon because you have antibodies and then they're giving you the toxin in a weakened form, right? And then you would have a really strong reaction at that injection site because it's too soon, right? You already have plenty of antibodies. Delayed cell mediated, could you get a skin reaction? Absolutely, that's what the TB test is, right? You can end up with an induration like you see with the TB test where it gets really hard, right? Or you can even have necrosis like what you see with poison ivy and poison oak and other contact dermatitises. It can be really gross. So this is that table from the book. As you can see, they have some different stuff on here, right? And I pared it down to what I wanted you guys to focus on. I didn't put my examples on here because we discussed it on a previous slide. So what are the antigens responsible for allograft rejection? So these are actually receptors on our surface that our white blood cells use to communicate with each other. And each person genetically makes theirs a little bit differently than other people. And that's why when we give you someone else's tissue, we're not going to be able to interact with this. This is your MHCs, right? Mainly your MHC class 1. Because remember, this is found on all your cells. This allows you to communicate with what cell type? MHC class 1 allows you to communicate with what other white blood cells? The what? B cells, right? Specific, no, not B. T, right? Which T cells? CD8, which do what? They're cytotoxic, right? So they are not going to be able to communicate with these cells, and because of that, they're going to kill them. So your antigen is something that is the antigens that create the problems, and this is where these guys got their name, major histocompatibility complex, right? This was the things on our cells that was creating the problem for organ and tissue transplants. Well, come to find out, the reason is because T cells are using this to communicate with our cells, right? And one of the main things they have to do is be able to recognize our actual MHC. So I actually added to your guys' blackboard, I don't know if you noticed it, we never really went over T cell development in the thymus, And I'm not going to test you on it, but it shows you, it's an animation that shows you about how they go through that process of being selected in the thymus, right? If they don't recognize MHC, they're going to be eliminated because they have to be able to recognize it, right, in order to be able to communicate with our cells, right? So that's one of the steps of the process, is actually making sure that their MHC can recognize CD4 or CD8, depending on which MHC we're talking about, 1 or 2. So what immune cells are responsible for the allograft rejection? It's going to be your T cells, right? CD8 T cells. What would happen if administration of anti-rejection drugs was discontinued? So when people get these transplants, they give them immunosuppressant drugs for the rest of their lives, because if they were to stop this drug treatment, their immune system might what? Recognize these foreign cells and kill them. Okay? They would die. Their immune system could recognize if it's not being suppressed and then eliminate the organ, and then therefore the person would die. This is when we say they're in rejection, right? So the major drawback is those differences, and it could be a type 4 type of hypersensitivity reaction, right, where it's those T cells that are recognizing it as being foreign. Natural killer cells too could get involved. They're the other ones. So we give immunosuppressant drugs specifically targeting those groups of cells.
So there's no way like you'll know like until it like happens like with a transplant. Of course, that makes sense.
In the beginning phases, when a person first gets their transplant, right, they'll be able to determine if it's going to take or not. But even during that time, they're taking immunosuppressant drugs. If that person was to stop taking those drugs, then their immune system could potentially recognize the foreign organ and destroy it. And the whole reason of getting it transplanted was because you kind of need it to live.
Yeah.
We don't get organs we don't need. So the other thing is too, that leaves you more susceptible to infection, right? So people who have transplanted organs, they don't go work at daycares, like little germ magnets walking around all the time. No, they'll stay away from crowded places, right? They're not going to festivals usually. Just too many people, too much chance of exposure to pathogens. All right, so we got to fly through this next one. So this one you don't have to worry about. Give 4 examples of autoimmune. All right, I'm going to eliminate that one. But the interesting things with autoimmune diseases may result from reactions to antigens that are similar to MHC. So remember how I mentioned before, like we naturally produce antibodies over time against the ABO blood groups we don't have? This is kind of a similar thing where, unfortunately, there are probably some antigens out there that have molecules on them very similar to someone's MHC. So somehow they produce antibodies against it. They don't get caught, right, that this is similar to a self antigen, and we start attacking our own cells, right? It's this kind of cross reactivity. Also, sometimes certain parts of our body aren't normally exposed to our immune system. Under injury, though, white blood cells might infiltrate into the area and see self-antigens as being foreign, right? Now, they were never deleted to begin with. So rheumatoid arthritis is an example of that, and I think that's what they're showing here in this picture, where people are attacking their own joints by their immune system. It could be antibody-mediated, it could be cell-mediated, it could be both. So this is some examples, right, where it's a specific example of what's happening. One of probably the most interesting one is T cells destroying people's beta cells, right, which are the ones that produce insulin. So this is what we refer to as type 1 diabetes. And this can be inherited, right, where unfortunately your T cells don't see your beta cells as being your own and attack and kill it. So this tends to develop early in life, unlike type 2 diabetes is a totally different type of situation. And then, as I said, arthritis is where immune complexes may be forming in the joints. You've got antibodies attacking those joints. You've got the immune system attacking those joints. As I said, I'm not going to ask you guys specific examples, so I'm going to gloss over that one for a second. Treatment in autoimmune disease, again, it's your immune system attacking self. So they're going to use different types of drugs to suppress the immune system. The good news for type 1 diabetes is a pretty easy fix, is that we just replace what the beta cells produce, which is insulin. Same thing with thyroid, like if someone loses their thyroid, we can replace the hormones that are associated with that. So there's two different types of immunodeficiencies. There is primary or congenital, and this is when you're born with a genetic defect where some component of your immune system is not functioning properly. And so there's different degrees of deficiency. Secondary or acquired you've heard before because it's part of the acronym that people say and they don't even know what they're saying, which is AIDS. AIDS is acquired immunodeficiency syndrome. At one time, they didn't know what was causing this, right? Why were people acquiring this immune system that is now deficient? That's why it was listed as a syndrome. We had these lists of symptoms. Nowadays, we know HIV, right? Human immunodeficiency virus is a very common virus that can cause the immune system to become deficient. You can be HIV positive and not have AIDS. AIDS means you've progressed to the level of HIV infection that your immune system is now deficient, right? And this leaves you susceptible to secondary infections. Most people who have AIDS, right, or HIV that progress to the level of AIDS, don't actually die from that. They die from not being able to defend against another infection, the most common being pneumonia, right? Because if your immune system is deficient, it can't defend, right? And so that leaves you susceptible to other types of infections. So again, I wouldn't ask you specific ones. One of your classmates was asking me about this one. I had mentioned it before. Severe combined immunodeficiency, commonly abbreviated SCID. This is a defect down at the stem cell level, right? So they don't have functional B cells or T cells. These are the bubble babies, right? Because they have a severely compromised immune system. Where something like IgA deficiency just leaves you really susceptible to stuff coming through your mucous membranes, right? You don't have that level of protection, but you have all the others. So as you can see, can vary. And as I said with SCIDs, right, it occurs in 1 in 500,000 live births, right? So most with this genetic defect they're not actually born, right? They die in utero, right? Because it's such a severe problem. Others experience where they don't produce antibodies or very few antibodies. Again, this is somewhat treatable by giving them someone else's antibodies. Right, and we've mentioned AIDS, but there are other types of infections that can cause you to have your immune system be deficient. Syphilis can really be a big draw on your immune system, so much so that you can become deficient. So can leprosy, malaria. Lots of other things can affect your immunological state. Remember, your immune system is your army, right? You've got to rest it, you got to feed it, right? And those things aren't easily obtained. Malnutrition, right? One of the first things that's not going to be maintained is that army, is your immune system. So then that's going to leave you susceptible to infection, right? As we advance in age, right, our immune system wanes. It's not as good as what it's supposed to be before, right? So environmental things, anything that's not genetic, right, would be secondary or acquired reason why your immune system is deficient. And I believe that's all I got for you guys, yep. That's it. So make sure you have, if you came in late, you read my announcement on Blackboard, I think it'll make you very happy.